Hi students, today I'm going to be giving you a brief introduction to immunity. I like to use an analogy that your body is very much like a castle. There are 3 layers of defense in your immune system, and I'm going to break each one down with that castle analogy. There's the first layer of defense, your chemical and physical barriers. Your second layer of defense is your innate immune system, and the third layer of defense is your adaptive immune system. So let's talk about those chemical and physical barriers. These are things that are the first line of defense because they're the first things that might interact with germs, bacteria, or viruses. So here you see tears. The tears in your eyes actually flush out and keep out things that might be bad for us. There are also some chemicals in those tears that help fight bacteria. We also have, of course, our skin layer. And that's a nice protective layer all over our body that prevents things like bacteria and viruses from getting in. We have our stomach and stomach acid, and that low pH is crucial for us to make sure that we break down any kind of bacteria that could be harmful for us that we take in through eating or drinking. Very lastly, we have mucus, and you might recognize that picture there of somebody blowing their nose, but that mucus that lines your mouth in terms of saliva, as well as your nose, that is actually a barrier. We excrete that to help protect our sensitive tissues from being invaded by bacteria and viruses and even fungus. When we think about that castle in terms of those protective barriers, really the castle walls and the moat are kind of equivalent to those protective barriers, right? These are the most external structures that protect us from invasion. So moving on to the innate immune system. The innate immune system relies on white blood cells. Now most of the time you think of blood as being red because we see the red blood cells, but there are also white blood cells in that bloodstream. There are a few specific kinds of cells that are white blood cells that particularly help the innate immune system. Those are neutrophils, macrophages, and dendritic cells. These cells are all phagocytes, and what that means is that they can engulf things like bacteria and fungi. This is actually a scanning electron micrograph of a macrophage that's phagocytosing a fungus, and that cell is going to eat that fungus in through this phagocytosis method and degrade it. There's a limited range of functions that these cells have. They mostly do this phagocytosis where they take things in and degrade them, but they can also release toxic mediators and chemicals, but they have a limited range of functions. One of the things that these cells are really good at is causing inflammation. And inflammation you might recognize as that redness and soreness that you get when you have a small infection. So sometimes when you get a cut and it gets a little infected, it turns red and it's sore and you have some swelling. Well, that's largely caused by these cells. Those cells are recruited to that area, and that's what helps with the swelling. They cause heat, and there's a lot of pressure buildup, and that kind of presses on our nerves and is what causes pain. So all of those things are part of our innate immune system. The reality is that innate immune system is very generic. There's a limited range of functions that those cells can perform. And so I like to think of the innate immune system as kind of rudimentary generic tools that you would have for any kind of intruder that was approaching the castle. You might use a bow and arrow to shoot arrows at something, or use a slingshot and sling rocks at something. But these are kind of generic, simple weapons that we might use for anything that's approaching the castle that we don't particularly like. So the adaptive immune system is a little bit different. We use different white blood cells. You can tell these cells on the right, and they look a little different than the cells in the last slide. These cells are responsible for fighting the infection in a different way. In particular, B cells are going to secrete antibodies, which I'm sure some of you have heard of. And T cells are responsible for activating those B cells, but they're also responsible for directly killing cells that might be infected with a pathogen. So B and T cells have these different roles. And one of the ways that it's really different than the innate immune system is that cells have to be clonally expanded, which means one particular cell will actually proliferate quite a bit. I'm going to use B cells as an example of clonal expansion, but this is true for T cells as well. So you make many kinds of B cells. So on the left side, this column here, you can see 7 different types of B cells. And each of these B cells has a slightly different shaped receptor on its surface. And again, we make millions of these. So in this example, there's just 7, but we make millions of these. Now, when we have an infection, we might see if any of the B cells we have would be a good weapon against the pathogen that we're encountering. Since each of these B cells has a different receptor, some of these receptors make that B cell good at fighting the infection, but most of them are not going to be good at fighting the infection. So it takes a while for us to figure out which B cell is going to best fight the infection at hand. The best B cell is going to proliferate like crazy, and that's that clonal expansion piece. So here in the center you see a B cell that's interacting with some kind of pathogen and getting activated, and then it's dividing. And then those cells divide, and then those cells divide. That's a clonal expansion. Only one particular B cell here was good at fighting the infection, and that's the one that proliferated like crazy. Now those cells, once they've proliferated several times, that's when they release their specific receptor, and that's what an antibody is. That released receptor is an antibody. And antibodies are important because they help trap and destroy pathogens. Again, I want to make sure that you realize that here in this picture I was using B cells, and B cells secrete antibodies. T cells also expand, and remember, T cells are going to help B cells, but they're also going to be able to kill cells of ours that might already be infected. So they have to go through a clonal expansion process as well, even though it's not shown here. And they don't end up secreting their receptor, but they do end up being able to interact with other cells and kill them if they happen to be infected. Adaptive immunity and clonal expansion means that millions of B cells and T cells are made each day. The appropriate cells must be identified, confirmed to work, and then expanded before things like antibodies can be released. So that takes time, and therefore this process is pretty slow. But in the end, it's worth the wait because the result is a tailor-made, specific response to fight an invader. So when the bow and arrow and the rocks aren't enough, we might need a very specific weapon to help us fight that infection. Okay, so the castle analogy for adaptive immunity is we're going to figure out which weapon from our large and ever-changing storage locker of B cells and T cells, we're going to figure out which weapon works, we're going to make a ton of that weapon, and we're going to use that to destroy the enemy. Those weapons are much more complex and exciting than the rock, slingshot, and bow and arrow, obviously. Here's a summary of some of the differences between the innate and adaptive immune system. The innate immune system is fast, but it's generic. It utilizes rudimentary tools to try and fight the infection. It uses phagocytes, white blood cells that can reach out and try and absorb and degrade pathogens. And it also uses inflammation, and that inflammation, remember, is heat and pain and swelling at the site to try and trap that infection so we can contain it and fight it locally. The adaptive immune system, however, is slow, but it is specific. It relies on special cells called B cells and T cells, which are also white blood cells. So let's review. The first line of defense against the pathogen is phagocytic cells, B cells, T cells, or skin, mucus, and tears. Pause it if you don't know the answer yet, because I'm about to give you the answer. Okay, the answer is D. The first line of defense is the chemical and physical barriers that prevent infectious agents from getting into our body, our castle. One more question. In order to have an adaptive immune response, you must... It's a real tricky one. Pause it if you don't know it. The answer is actually both A and C are true. This is for the most part. For most infections that you're ever going to have, you actually are going to need to clonally expand B cells and T cells in order to get the best immune response. Okay, well, I hope you learned something about how the immune response works, and we look forward to talking to you more about this in class.
In this topic, we're going to discuss the principles of immunity, antigens and antibodies. So we're going to look at what is an antibody and antigen, the features of an antibody, the structure of an antibody, the difference between self and non-self cells, the functions of antibodies, the classes of antibodies, and then I'm going to give you a little bit more information on monoclonal antibodies that are used in pregnancy test kits. Immunity is the ability of organisms to resist infection by protecting against disease-causing microorganisms that invade their bodies. So it involves the recognition of foreign material and the production of chemicals that help destroy it. So here you can see a pathogen, and on the surface of the pathogen are little proteins called antigens. So any molecule that the body recognizes as foreign, it's usually a protein, will initiate antibody formation. So this is called an antigen. The antibody, which is represented by the orange, is a protein called an immunoglobulin, which is made by B lymphocytes in response to these antigens. So the immune response is when these B lymphocytes respond to the presence of antigens by secreting antibodies. So let's have a look at antibodies in a little bit more detail. Antibodies are also known as immunoglobulins. They're proteins synthesized by cells in the blood by B lymphocytes. So when the body is invaded by foreign material, so an antigen, B lymphocytes produce antibodies that are going to react with the antigens on the surface of that pathogen. So antibodies are very specific to the antigen. As you can see on this antibody, you've got 2 sites at the end of the antibody where the antigens can bind. So antibodies comprise 4 polypeptide chains. You've got 2 heavy chains and 2 light chains. So notice how the heavy chain is in the middle and the light chains are those smaller chains on the outside there. Okay, let's label the different parts of the antibody. The yellow part at the top there represents the antigen binding sites. This means that the antibody can bind to 2 antigens at the same time. This is also called the variable region. The chains are held together by disulfide bridges, and they link the polypeptide chains and maintain the quaternary structure. The hinge region, represented by those dark blue circles, is flexible, so it gives flexibility when the antibody binds to the antigen. The constant region is the same in all antibodies, and it's where the antibody binds to a receptor on a macrophage. Now, to be able to defend the body from invasion by foreign material, B lymphocytes must be able to distinguish between the body's own cells, which you can see on the right, and chemicals on the left. So the body's own cells are called self cells, and foreign cells are called non-self cells. So here you can see a B lymphocyte travels around the body. It comes in contact with a self cell, recognizes it as its own from the own body, so it doesn't do anything. Then it comes into contact with a non-self cell and it binds to it, and then an immune response will be initiated. Now, if the B lymphocyte didn't recognize the body's own cells, it would produce antibodies that would destroy the organism's own tissues. So how do B lymphocytes recognize their own cells? So here you can see the B lymphocytes moving around the body, and they come into contact with self cells. They recognize them as their own body cells, so they leave them alone. Now, each antibody is produced by a different lymphocyte. In the fetus, the lymphocytes come into contact with their own cells. Infections in a fetus are quite rare because it's protected from the outside world by the placenta. So as I showed you earlier, the lymphocytes will collide with almost exclusively their own material, so self cells. Some lymphocytes do have receptors that will match the body's own cells. So you can see that represented by this light blue B cell. These lymphocytes will be selected against or destroyed. so that the only remaining lymphocytes are those that fit foreign material, so that the antibodies that are secreted will only be against foreign material and not against the body's own cells, the self cells. Let's have a look at the functions of the antibodies. The functions depend on the type of antigen to which the antibody binds. So the first is the antibodies can combine with viral or bacterial toxins and prevent the entry of these into host cells. They can attach to the bacterial flagella and immobilize the bacteria so that they are easier to engulf. They can cause agglutination of bacteria by attaching to multiple binding sites. They can work with other molecules to make holes in the bacterial cell wall so that water can enter into the bacterial cell, and because the bacterial cell wall is now weak, the bacterial cell will burst. They can coat the bacteria, providing recognition sites for phagocytes. Or they can act as antitoxins by combining with and neutralizing toxins. So here's another diagram to show you the different functions of the antibodies. So these include neutralization, agglutination of microbes, precipitation of dissolved antigens, and making holes in the bacterial cell wall. Right, let's move on to the different classes of antibodies. You've got IgG, which have got 2 binding sites. They're found in the blood and tissue fluid, and their function is to enhance the activity of macrophages, to act as antitoxins, and to cause agglutination. IgM have got 10 binding sites. So you can see that there are 5 antibodies, and each has got 2 binding sites, so it's 10 binding sites in total. They're found in the blood and tissue fluid, and they initiate the primary response, and they also cause agglutination. IgA have got 2 or 4 binding sites. They're found in your saliva, bronchial secretions, mucus secretions, and also in colostrum. So they inhibit bacteria binding to host cells or forming colonies on mucous membranes. And then lastly, you've got the IgE, which have got 2 binding sites. So they're found in your tissues, and they activate cells to make histamine. So they are involved in allergic responses to harmless compounds. So if you have hay fever, it's your IgE. Okay, a little bit of extra information on monoclonal antibodies. Now, when a pathogen invades the body, this pathogen has got many different antigens on its surface, and each antigen will induce a different B lymphocyte to multiply and clone itself. So you have a mixture of different antibodies being produced. In medicine, it's beneficial to produce a single type of antibody from just one clone of B lymphocytes, rather than having a mixture of antibodies. So these are known as monoclonal antibodies. Mono meaning one. So monoclonal antibody is an antibody produced by a single clone of cells or cell line, and it consists of identical antibody molecules. So where would we use monoclonal antibodies? Well, in a pregnancy testing kit, what you do is you've got immobilized antibodies on something called a urine dipstick. So this detects traces of a hormone called HCG, human chorionic gonadotrophin hormone, that gets released by the placenta. So if we look at the different stages, you can see that antibodies are tagged with a particle. So the antibodies are tagged with a particle, represented by those green drawings there. The HCG binds to the first antibody that's got a colored particle attached to it. This antigen-antibody complex then moves along the dipstick until it reaches the test zone, where it comes into contact with immobilized antibodies that are represented by the blue. These bind, and this is where you get your first line in the pregnancy test kit. The antibodies that aren't attached to the HCG then attach to the next zone of immobilized enzymes. This is the control zone and forms the second line in a pregnancy test kit.
Let's start our discussion about immunology. Immunology is science and study of an organism's immune system. So what is our immunity? Our immunity is our body's protective mechanism against pathogens. Pathogens being disease-inducing organisms or viruses. Why do we need an immune system? We need it essentially for living, because we're constantly attacked by different pathogens, microbes, and anything essentially that could potentially harm us. When is our immunity applicable? It's applicable throughout our life, from the day we're born until the day we die. And how is the mechanism? This is the discussion that will take the longest time. So let's start with our lines of defenses. We have 3 lines of defenses: first, second, and third. Our first line of defense will be our passive barriers, in which we have our skin, our mucous membranes, our stomach acidity, which will inhibit a majority of different pathogens entering our system. Remember that the pH in our stomach is around 2, so it means it's very acidic, and it gives a very unfavorable environment for different microbes to survive. Then we have our skin cells that are tightly placed next to each other, inhibiting a lot of bacteria and viruses entering our system. If the pathogens would be able to pass our first line of defense, we have our second line of defense, which will be our innate immune system, in which we will have different cells and proteins that would be non-specific to the different pathogens and try to kill as many of them as possible. The complement response will be essentially our complement pathways, mainly the lectin pathway and the alternative pathway. The phagocytes will include our neutrophils, our macrophages, and our natural killer cells. Granulocytes are also the neutrophils, eosinophils, and basophils. Granulocytes refers to that they have secretory vesicles. If the pathogens would be able to pass the second line of defense, we have our third line of defense, in which our adaptive immune system will play the majority of the function. In here we have our lymphocytes, which are our B cells and T cells. So where do we get our cells? All of our cells are derived from different stem cells. Stem cells are essentially blank cells, meaning that they are completely undifferentiated. And as they differentiate more, they mature more and become very specific by its function. For when it comes to our blood cells, we have our hematopoietic stem cells, which is found in the red bone marrow, in all of our bones in childhood, but when you're an adult, your skull bones, your vertebra, your ribs, your sternum, your scapula, pelvis, and the proximal portions of your humerus and femurs are working, while the rest of your bones has converted into fat called yellow marrow. So these stem cells can differentiate into two lineages, and this will depend on which chemical messenger they will release. If they would release the cytokine interleukin 7, they would go towards the common lymphoid progenitor, in which you will find your lymphocytes and natural killer cells. But if they would release interleukin 3, then they would go towards the myeloid progenitor cells. In the myeloid progenitor cells, we have most of our blood cells, our erythrocytes, also known as the red blood cells, our platelets, also known as the thrombocytes, and most of our white blood cells, such as our granulocytes, that are the neutrophils, eosinophil, and basophil, and also one of our agranulocytes, that is the monocytes, later formed macrophages. Another agranulocyte is our lymphocytes. We have also different lymphoid organs. This will be the site of production of blood cells and also maturation of some white blood cells. So first we have our primary lymphoid organs, in which that are the bone marrow and the thymus. Bone marrow, as mentioned before, is the production site of all blood cells. Thymus is the maturation site of your T cells. T cell stands for thymus cells or thymocytes. But remember that the thymus will, prior to your puberty, convert into fat behind your sternum and will be called retrosternal fat tissue. And its role will be taken over by your secondary lymphoid organs. These include your lymph nodes, your mucosal associated lymphoid tissues, your spleen, Peyer's patches in your intestine, and so on. These have another function of bringing your different antigens. These are the binding sites of the different pathogens to this area and expose them to your lymphocytes for their function, for their later function in the adaptive immune system. So what is the biggest differences between your innate system and adaptive? Innate system is also called the non-specific system, and therefore it's very unspecific to different pathogens. So it will essentially kill whatever it resembles as foreign, while your adaptive system will be quite specific to the different pathogens that are exposed. Another difference is the diversity. This refers to the range of its action. The innate system has a narrow range of action, meaning that it can only neutralize certain amount of pathogens. But your adaptive system, due to its somatic recombination, it has a wide range of actions and wide range of pathogens to neutralize. But one of the biggest differences between the innate and adaptive system is its memory. The innate, as it's non-specific and narrow-ranged, it does not have a memory. So it does not remember whatever it has been exposed to before. But your adaptive system will remember what it has been exposed to before and be more prepared next for the second exposure. Both of these systems, the adaptive and immune, can become pathological and start to attack your own body cells. And when it comes to the physical and chemical barriers, these refers essentially to our lines of defenses. The blood proteins in the innate system is the complement, but it's not all of the complement pathways. Only the alternative and the lectin pathway are involved in the innate system. But the classical pathway of the complement system requires an antibody, and therefore it's associated with the adaptive immune system. When they write, what you see here written about antibodies, they refer to the humoral response, which we'll talk about later on. And we mentioned the cells associated with the different immune systems. Remember, all of these cells will be spoken about individually in upcoming videos. Thank you.